So transport in plants. So as for transport, from where it should take place, transport usually takes from the roots or sometimes from the leaves also, the transport mechanism, but especially aerial parts. The aerial parts of the leaves, mainly as the plant is concerned, the transport mechanisms which takes place either from the roots or from the aerial parts of the leaves. What are things will be mainly get transferred? The water, your minerals, your nutrients, you take the organic nutrients. These compounds like enzymes, the growth regulators, usually which is getting transferred. Okay, so how it is getting transferred? By means of the specialized vascular tissues, we call it as through the xylem. So by means of xylem and phloem. So what the xylem transfer and what the phloem transfer? So xylem usually transfer the water molecules. Xylem transfers the water molecules, and phloem usually transfer the organic solvents. Usually transfer the organic solvents. But xylem has nothing to do with the organic, but only the phloem which has the ability to transfer. Priyanka, I couldn't see your message. What you said? Hyponasty means downward. Yes, yes, Priyanka, right. You're correct, Priyanka. Yeah, fine. Priyanka, correct always. Okay. Phloem for the organic solvents like the minerals, but in xylem only the water which will be getting transferred. Fine. Now we move on to the means of transport. Guys, we're not going depth in this chapter. I'm just giving only the outline. Okay. Means of transport. As far as the means of transport is cellular level, shorter distance, and the long distance. There are three types of transport. Cellular level transport, shorter distance, and long distance transport. What is the cellular? Within a cell. Within a cell, how the transport mechanism is going to occur. Shorter distance between one cell. This is for cell A. This is a cell B. How the transfer is going to occur between the cells. That is the shorter distance of the transport. And long distance, like from the root and to the aerial parts to the remote regions, like the transpiration pull on all the mechanisms takes place. That is the long distance. In other means of transport, you would have studied this takes place by means of active transport and passive. Guys, these are the common things. I'm not teaching again because you studied already this chapter. So I don't want to waste your time again by teaching right from the beginning. These are the basic things that you know already, right? So active transport and the passive transport. You know the difference between the active and passive transport. In active transport, definitely it requires some energy. So energy is requires a form of ATP, and this requires high concentration of GLUT. What is GLUT? Can someone tell me? It requires ATP, that is like energy currency of the cell, and this is a GLUT. For longer distance, you take Kavya said like an ascent of sap. Yes, exactly. You said ascent of sap, addiction, cohesion. That we'll be discussing in this part. The long transport. Yes, Kavya. Good. Ascent of sap. So active, you require the ATP, and this requires GLUT, GLUT. That is a glucose transport which is required. But coming to the passive transport, so types of passive transport, we have a diffusion, and we have a facilitated diffusion. We have facilitated diffusion, and we have the diffusion pressure. We call it as a DP. So we have the other mechanism called as diffusion pressure. So there are different three types of the passive mechanism that you could see. So coming to the normal diffusion, but in this part, the diffusion where it doesn't require energy transfer, no energy transfer, no ATP is required for this process. In the normal diffusion, what it takes place? Usually the gas, it takes place, maybe the gas, it takes place in liquid, it takes place in the any form, the gas liquid, and where the concentration which moves from what? From the higher concentration to the lower concentration for the passive transport. But in this case, like from the lower concentration to the higher concentration. This the difference between active and the passive. Here it moves, the molecules moves from the higher concentration to the lower concentration. The active it is lower concentration to the higher concentration. But diffusion is the slow process. Diffusion is a, compared to the active, diffusion is the slow process and is not much specific. But whereas like active process is very specific in nature, diffusion seems to be, it is a slow process, and this requires kinetic energy of the particles. This requires what? Kinetic energy. It requires kinetic energy of the particle. Okay, so the diffusion slow process, and it requires kinetic energy of the particles and difference in areas of concentration of the particle that you could see. But you could see in DP, that is like the difference in pressure, the diffusion pressure. This is exerted by how the diffusion pressure occurs. This is by means of the pressure exerted by the particle, and it is proportional to what? Proportional to the kinetic movement of the number of diffusing particle. And when you see the diffuse pressure, the value at 20 degrees C. When you see the diffusion pressure at the value, you could see at 20 degrees C, and it is not depend on the living system. Exactly. Suhana Yasmin is exactly that. She also given a very correct answer. At stop, Suhana. You also gave a valid answer. That is, it's not depend on the living system. That is also very good. Okay, fumigation. You know fumigation. Where do you apply fumigation? Usually, for example, you want to in operation theaters, any places that you go for a fumigation. You want to sterilize the rooms, or even in cinema theaters or closed environments, you go for fumigation. Even fumigation is a common thing that we apply the perfumes. So you apply the perfume, right? What mechanism is that? You apply perfumes on your body, what is actually, is it like diffusion or any type of other mechanisms? Or you just place a drop of ink in a beaker which consists of water. What mechanism is that? So if you just place a drop of ink in a beaker, it spreads inside. What type of mechanism is this? It's a diffusion. It's a common thing. It's you said in a fumes. So Shanmugam, you asked me fumes. We asked fumes as an example. Example for what type of reaction? It is like diffusion or facilitated diffusion is again the diffusion mechanisms. Get it? So incineration. If you burn something, you're burning, right? Fumigation is fine. You said fumigation. You're incinerating. What is incineration? Burning mechanism. For example, dead bodies or something like corpses that has to be burned immediately means you're burning it completely, right? What is the type of the process here? Can you say it's a diffusion, or what is the mechanism behind burning something or incineration? Fumigation is a diffusion. How about incineration? So dp is a diffusion pressure. This is proportional to the kinetic moment and number of diffusing particle, and the value at the 20 degrees centigrade is 13336.40 atmospheric pressure. The diffusion pressure, the value of the diffusion pressure at 20 degrees C is 1336.40. 1336.4. The transpiration causes water level rise up due to what's an active or passive transport. It depends upon the transpiration. For example, the transpiration mechanism, which is again a shorter distance or the longer distance. If it is again shorter distance, it is a passive process. If it's going with a longer distance, it goes to the active. Then you could see the activation pull, right? You know the transpiration is a necessary evil. In that case, of course, the plant has to spend an energy. If it is a long distance, definitely the plant has to spend the energy, that is ATP. It comes under the active process. It all means the distance. Distance. Formalin, and which produce enormous fumes which kill the pathogens, is enclosed area, and a method is known as, yes, exactly, fumigation. Exactly, usually that we do in case of sterilizing the closed places. For example, any contagious persons, you want very much concerned the disease should not get spread to the other person. You often you sterilize the rooms, right? Operation theaters and all. So by means of fumigation. Okay, depends upon the water surface. Exactly, Kodai Priyanka. Yeah, it depends upon the water surface whether it's a shorter distance or the longer distance. It depends. We cannot confine. It should go only with the active transport or only with the passive transport. It again depends upon the distance and the surface. Okay, the water surface. Fine. Okay, so we're done with the diffusion pressure. Facilitated diffusion. Here also the diffusion is going to take place. There's no expenditure of the spending of energy, but it requires some, the channels. By means, these proteins are going to facilitate the reaction. Diffusion is going to get facilitated by means of this particular, the proteins which going to be facilitate the diffusion, which going to enhance the reaction. This is called as facilitated. This facilitated reactions can be called as two types. When we talk about the facilitated, there are two types of facilitated diffusion. One is carrier type. There are two types of facilitated diffusion. Guys, maybe the board seems to be clumsy. Please, because I'm not erasing it up. Because if I erase you just miss the other part. So I'm writing the same board. I'm writing here facilitated diffusions of two types. One is your carrier type, and the other type of the facilitated diffusion is a channel type. We have two types of facilitated diffusion. What do you mean by the facilitated diffusion? Here, the diffusion is going to get facilitated or enhanced. How? By means of these one, the carrier, and by means of channel. So carrier, where you could see the carrier proteins will be there. This proteins which just carry the molecules from the outer surface and bring it inside. It facilitates the reaction, but no expenditure, no spending of energy in this case. right? And channel is actually, you could see in case of porins and aquaporins. The channel proteins, we call it as, we have two types of channel proteins, porins and aquaporins. Porin and aquaporins. Coming to the porins type, this like pore-like structure, where you could see this porins, you could see in case of mitochondria, chloroplast, or you could see in the bacterial membrane. You could see in the bacterial membrane, these porins. And coming to this part, the aquaporins, usually these aquaporins are 8-unit structure. This consists of 8 units, aquaporins. These 8 units, okay, these 8 units together, okay, which fuse together and form a water channel. This forms a water channel. 8 units of aquaporins which fuse together and form the water channel. So by this, the channels can be classified into porins and aquaporins. Understand the facilitated diffusion is the normal type of diffusion, but the diffusion mechanism is going to facilitate it by means of special type of proteins. I mean, the diffusion is going to enhance. How? By means of the carrier protein or by means of the channel protein. The carrier proteins which is present on the superficial surface, which receives it and enhance the diffusion reaction and bring it inside, and this carrier protein which go again to the extrinsic surface. Channel proteins that you could see, which is two types, porins and aquaporins, right? And for us, aquaporins are concerned, this is usually 8 in number, and these 8 units which club together and form the water channels. Okay, so when you talk about this particular, usually this porins, where you could see the mitochondria, chloroplast, you could see in the bacterial membrane, all these places we could call it as a thing. Okay, porins. So how we can differentiate the porins, where you could see exactly the porins? Can someone tell me the difference between the porins and aquaporins? If I tell you like an aquaporins, we call it as a water channels, right? And what is the main the function of the porin? Is there any particular, because we have the porins are concerned, where porins are concerned. For example, this is a surface. You could see here. You could see the porins actually. This is how the porins will look like. So the porins something like embedded proteins. You could see here the molecules like which will be passing through inside. This is the porin that we're talking about. This is actually the porin talking about, and this will be guiding this particular the molecule to get inside. Actually, this facilitate the movement of what. This will not move. The porins will not move. What this will do is this will facilitate the movement of this the molecule to come from outside to inside. So this type of facilitation which takes place by means of a porins, and this is the large transport proteins which usually present in what, in the usually present in the outer membrane. Where exactly that you could see this type of proteins, I mean the porins, you commonly could see in case of I think it's in plastids. You could see in case of plastids. Okay, and here it is not actually, we're not inducing it, we're not forcing it. It's happening, taking place, just spontaneously it is taking place by means of this protein, but it's not smaller, but it is like larger when compared to the other type of porins. Okay, but this is also like enhancing, facilitated type of diffusion. All right, guys, so we're done with this part. And osmosis, you take osmosis and you take plasmolysis. Even this comes under the passive type of transport, type of transport. So we just for 2-3 minutes I try to close and you can go for a break. So the next part that we discussed is about the types of transport. You would have studied about uniport, symport, these 3 types that you would have studied, right? So uniport, symport, and we have a antiport. So guys, 2 minutes, okay? Bastina Mary, just 2 minutes. Uniport, where the molecules, there's only one molecule which moves in one particular, because there's only one molecule here. You could see in symport, what happens in symport? Here the 2 molecules in the same direction. Here you could see the 2 molecules which moves in the same direction. But in antipodes, we have the 2 molecules which moves in the opposite direction, So opposite direction that you could see. So this we call it as an antipode. So this is the different ways by which the types, the molecules which can move. What are the advantages of diffusion? I hereby close, guys. Don't worry. Advantages of diffusion. When we talk about advantages of diffusion, so what are the advantages? Which keeps the cell wall moist, because the diffusion is taking place, like it keeps the cell wall moist almost all the time, and also responsible for gaseous exchange. Gaseous exchange during what? During photosynthesis. During photosynthesis, the gaseous exchange takes place. For that, diffusion is quite beneficial, and also for respiration, transpiration, and also you take in case of translocation. Also in case of translocation, that is translocation of or translocation of usually food materials. It also plays a very important role in translocation, translocation, and photosynthesis. In all these cases, the diffusion is playing a very important role. Okay, so this we done with the diffusion process, and the next part we'll discuss about osmosis and the remaining part about the transpiration. So now the time is 3 2 guys, so we go for a okay, Kriti, we go for a break just for 5 minutes, just for go for a break and just come back soon. Okay, have a break. Welcome you guys back after the break. Fine. So we are at the pathways that you could see uniport, symport, and antiport. And you ask about like symporter, right? So I could see the comment here, the symporter. So what is a symporter? It's symporter. Usually we call it as symporter. We call it as a co-transporter. So we call it as co-transporter. Symporter can be also called it as co-transporter. This is the other name that we give. Where this importer is present, this is present as the integral membrane protein. Usually this integral membrane protein which is present in the plasma membrane. Integral membrane protein. IMP is integral membrane protein which is present in the plasma membrane. And this importer which also we call it as a transporter. Fine. So this is what we asked been, and we also about the ROS. ROS, guys, you already given the answers for ROS. That is relative oxygen species. What happens in relative oxygen species where there will be the loss of electrons takes place? The leakage. I mean the leakage of electrons, which will be taking place onto the oxygen. Okay, from the electron transport action. So this is what we done so far. I mean uniport, symport, and antiport. Next, we move on to the osmosis. So coming to the osmosis, what type of transport is this osmosis? It's active or passive. So this osmosis you'll study in a you would have performed that in a practicals also the osmosis the experiment that you performed using a potato, right? Coming to this process, who proposed the osmosis? You ask guys like any questions comes from the discovery part from the year or this area you get a question. Yes, you get a questions from the discovery. So that is the reason like often anything that we take historical perspective, the scientist and the year of invention also we're giving importance for this area. So as far as the transport and plans is concerned, you get like 3 to 4 questions you get from this particular chapter. Coming to the osmosis, as far as osmosis is concerned, who first proposed osmosis? Proposed by Knowledge. Knowledge in the year 1748. So 1748, the Knowledge is a passive type of transport. How it depends upon? It depends upon the pressure gradient and the concentration gradient. It depends on pressure gradient, pressure and concentration gradient. And how it occurs by means of the semi-permeable membrane. So you could see the semi-permeable membrane. You could see in this case like the salt water, the fresh water. So can you tell me like the mechanism of the reverse osmosis? We call it as like RO water. So what is the mechanism? Is it like same mechanism like an osmosis? Or is any manipulated technique that you could see we get RO waters, right? Reverse osmosis mechanisms. What type of mechanism that you could see in a RO, that is in reverse osmosis, where the principle remains same as like that of osmosis, or is there any manipulated technique that you could see in case of RO, that you drink RO waters, right? In that case I'm talking about. So it is a passive mechanism. So again, this is the osmosis is a passive mechanism. So this depends upon, I told you, like pressure gradient and the concentration gradient. So there is something called it as osmotic concentration. So what do you mean by osmotic concentration? So osmotic concent, yeah, this is osmosis. So coming to the osmotic concentration where the amount of dissolved solutes that is per unit volume of the solution. That is what actually we call it as osmotic concentration. So osmotic concentration, the definition is what? Amount of dissolved solutes per unit volume of the solution. So this can be classified. So how this osmotic concentration can be classified? It can be classified into hypertonic, or it may be hypotonic, or it is isotonic. Hypertonic, or isotonic. So pressure gradient is applied in reverse through the semi. It occurs in only the pressure gradient. Actually, the pressure gradient is applied in the reverse manner actually by using the same membrane, that is semi-permeable membrane. That only makes a difference with the reverse osmosis. Okay. Out of 1826, guys, you said like the year is concerned, so let us not waste most of the times because the reference shows 1748. But you get confirmed with 1826. I need a reconfirmation from your side. Okay, we'll see to that. Okay, it takes place in selective permeable membrane at reverse osmosis. Okay, so hypotonic, and isotonic. So what happens like in hypotonic where the osmotic concentration is greater? So what happens like osmotic concentration. Osmotic concentration is greater. Greater than what? Osmotic concentration is greater than the cell sap. Is greater than the cell sap. It is hypertonic. What is hypertonic? Here osmotic concentration is lesser than that of the another solution. Then in isotonic, both the cell sap as well as the solution where you could see it is equal. It is equal. So the osmotic concentration can be classified into hypertonic, hypotonic, and isotonic. But when you're talking about the types of this osmosis, we have exoosmosis and endoosmosis. Exoosmosis and endoosmosis. What makes the difference between the exoosmosis and endoosmosis? What means into the RO water molecule move from the lower to the higher potential. Yes, Kirti, from the lower to the higher potential. I solute, that is hypertonic is the I solute. Okay. It's a passive process which comes under the passive process. Passive. So can, if you like, justify, is any places where we confirm like osmosis as an active process, usually living organism, living, like, plasmolysis is concerned, like in plasmolysis or in osmosis, can you give any confirmation that these osmosis and plasmolysis is an active process? Usually we won't consider this an active process. It is a passive process only. It's a passive process. Okay, fine. Fine. So we're done with the types of osmotic concentration: hypertonic, hypotonic, and isotonic. And the difference that exoosmosis and endoosmosis. Exoosmosis, where what happens like exoosmosis? Cell in a hypertonic solution. So usually the cell in a, you can place the cell in a hypertonic. You can place the cell in a hypertonic. Then okay. Coming to the endoosmosis. Endoosmosis, where we can keep the cell in a hypotonic solution. Endoosmosis, we keep a cell in a hypotonic solution. If you keep a cell in hypotonic solution, what will be the fate of the cell? So it may be like turgid or it may be a flaccid. Flaccid. So where it will be turgid and where it will become flaccid? If you keep in exoosmosis, if the cell is placed in hypertonic solution, definitely the cell will become flaccid. Here the cell will become flaccid. And if you keep the cell in hypotonic solution, the cell will become turgid. So this is the fate where you keep the cell in a hypotonic solution. In case like if you keep the cell in hypertonic solution, the cell will become a flaccid condition. Okay, flaccid condition. Okay. So this is about the osmosis where you can differentiate exoosmosis and endoosmosis. Coming to the osmotic pressure. Osmotic pressure. I write it here. Osmotic pressure. Osmotic pressure can be denoted by pi. It can be denoted by the symbol pi. Okay. So what is actually osmotic pressure? It is actually pressure in a actual pressure. It's a turgid. Turgid in a endoosmosis and flaccid. Flaccid in case of exoosmosis. Again I tell you, when you keep the cell in a hypotonic solution, what happens? The cell will become turgid. Okay. When they keep the cell in a hypertonic solution, then the cell will become a flaccid. Very in detail, we discuss about the plasmolysis. What's the indication like, when the cell goes to the turgid, that we'll discuss in detail in plasmolysis. Coming to the osmotic pressure. This is the pressure in a solution where what happens here, it's separated from its pure solvent. Pure solvent is water. It is separated in a pure solvent. Here the pure solvent is used as a water by means of what? Here using a semi-permeable membrane. Okay, use a semi-permeable membrane. What is osmotic pressure of a plant cell? Can someone tell me what is the osmotic pressure of a plant cell? What is osmotic pressure of a plant cell? One sec, guys. What is osmotic pressure of a plant cell? So coming to the osmosis, like the questions they ask usually with the principle based, you ask, you may get a question, or you get a questions like the semi-permeable membrane, or they ask from the pressure, osmotic pressure of a plant cell. If the osmotic pressure of the plant cell, the range is like 4 to 5 atmospheric pressure. 4 to 5 atmospheric pressure is the osmotic pressure of the plant cell. Okay. One more thing, osmotic pressure is directly proportional to the concentration of the solution. So this is directly proportional. Osmotic pressure is directly proportional. It's directly proportional to both the concentration of the solution and to the temperature. Concentration of the solution and the temperature. In this area also, from the osmotic pressure, you can ask a question. The question comes like, which among the following is the correct statement? Where you could see osmotic pressure is directly proportional to the concentration of the solution and the temperature, and osmotic pressure of the plant cell is 4 to 5 atmospheric pressure. Fine. So we're done with the osmosis. Then we'll see about plasmolysis. When we come to the plasmolysis, so here in this place you could see plasmolysis. There are three types of plasmolysis that we discuss. Plasmolysis, the plant cell is placed in a hypertonic solution. If placed in a hypertonic solution, definitely what will happen if a plant cell is placed in a hypertonic? So this is the hypertonic solution I'm talking about. I write it here. The plant cell which plays in a hypertonic solution. So what will happen? What will be the fate of the cell? What will be the fate of the cell if you place a cell in hypertonic solution? Then exoosmosis takes place. If exoosmosis takes place, then what happens? Definitely the water will move out. The water will move outside. If the water is moving out, then what happens? Definitely the protoplasm will start shrinking. So this will shrink. Protoplasm will start shrinking. Okay, this condition is called as plasmolysis. This is called as plasmolysis. What is the condition for plasmolysis? If you keep the cell in hypertonic solution, okay, where you could see in case of where exoosmosis takes place, where the water is going out of the cell, then automatically the cell will become what? It will become shrink, and definitely it goes to the plasmolysis. Based on which, right? Yeah, flaccid. Yeah, correct. Where the plasmolysis, which can be of three types actually. What are the three types? Where you could see in this, like, it is turgid, where the water is coming inside. So in this diagram, you could see the reference here. The water osmotic pressure which is coming inside, it is turgid. When equilibrium condition is maintained, so equilibrium condition is maintained, the cell is in isotonic conditions where the like exchange of water molecules in the equilibrium state. But water loss takes place. Water loss takes place, then again what happens? Like, it leads to a plasmolysis, and the cells will start shrinking. So based on like the size of the protoplasm that you could see, especially in the case of plasmolyzed cell, okay, what are three types of the plasmolysis? The three types is limiting, incipient, and evident. Limiting, incipient, and here it is evident. There are three types of plasmolysis. Coming to the initial, yeah, good, you were straight. Limiting, plasmolysis, incipiting, and evident. Good. In the limiting, where you could see like turgor pressure. What is the turgor pressure for a limiting? Can someone tell me what is the turgor pressure? The turgor pressure for the limiting paralysis. Limiting plasmolysis. Incipient. What is the turgor pressure here? Turgor pressure. The turgor pressure for evident plasmolysis. Here you could see the change in the protoplasm. In the initial stage where you could see the limiting, where you could see now you don't see much of the difference in case of the protoplasm. Here you could see. And later part you could see the difference. You could see the difference here. Now the cell starts slightly got shrinking, and now the protoplasm, the vacuole size also getting reduced. At last, what happens? Like protoplasm like size is getting completely shrink inside. And this cells we call, this is the evident. The last type is called as evident plasmolysis. This one is the evident plasmolysis, where the vacuole size got reduced and the protoplasm it's almost got shrink. And these two stages, we call it as incipient. This is the incipient plasmolysis, and this one is a limiting plasmolysis. Limiting plasmolysis. Based on the size of the vacuole and the protoplasm, again, this is getting, is this occur to human being long in a sea? Okay, this is quite of course, this also, you could see in case of the most of the cases where in human beings also. Okay, fine. One second. Okay, fine. Okay, guys. Okay, this is a process that you could see in case of the human beings also. Fine. So plasmolysis, three types where you could see the limiting, incipient, and also you could see evident. Okay, there's three types of thing that you could see. So what's the question, Shanmugam? Can you ask the question? So Kodai Priya, I tell you like plasmolysis are three types. We have limiting plasmolysis, incipient plasmolysis, and the evident plasmolysis. Okay, this you could see the plasmolysis where you could see the limiting plasmolysis where the plasmolysis is about to takes place. Well, what happens like you won't be seeing much of the radio reduction size of the vacuole, not much of like reduction in the size of the vacuole or like size of the protoplasm. It's not about to get strings. It's a initial stage, incipient stage, where it's like slightly what happens like from the cell wall. What happens like it's the protoplasm and the vacuole will start reducing its size. This is called incipient. The vacuole will start reducing in size. But at the last stage, the evident plasmolysis, what happens like the completely like the vacuole will start reducing. It started reducing. So this we call it as a plasmolyzed cell. Okay, guys. So the turgor pressure, when you talk about the limiting plasmolysis, the turgor pressure is zero. Turgor pressure is zero. And coming to the incipient plasmolysis mostly it is negative. Incipient plasmolysis is negative. Evident plasmolysis, it is more negative. It is more negative. So, but the turgor pressure in the initial stage of plasmolysis, it is a zero. So this is just a brief introduction, I mean like outline of the plasmolysis. Next, we move on to the imbibition. So what do you mean by imbibition, guys? Can someone tell me what is imbibition? Imbibition. Coming to this part, the imbibition. So what is imbibition? Limiting and incipient in the diagram. So what is the stage? That is a different stage of the plasmolysis. The initial stage of the plasmolysis and the last stage of the plasmolysis. It depends upon the changes that are going to take place. Change takes place in the reduction size of protoplasm and the vacuole. So eventually the cells start shrinking. So that is based on that we differentiate into three. The swelling due to the water gain. If there is swelling, then what happens is because of water gaining. If the cells start shrinking, then what happens? There is a loss of water. Okay, fine. The limiting plasmolysis, but given the final, there are three types of plasmolysis. So there are three stages in the plasmolysis. So you can refer the textbook once again. So in the plasmolysis, they're given limiting. Limiting is the starting stage. Incipient stage is the middle stage where the turgor pressure is negative. Evident plasmolysis where you could see more negative, where you could not see the much of the vacuole formation and also the protoplasm start reducing. And the imbibition, what was observed that you see like solids and increase in the volume. Okay. Jennifer, I couldn't see your question, Jennifer, because your comments gone up. I couldn't see your question yet. Okay, fine. If I could see, I answered your question, Jennifer. Okay, fine. Okay. So coming to the imbibition type. As far as imbibition is concerned, this absorption of water by a solid particle. This is absorption takes place by means of absorption of water by solid particles. Usually it occurs by means of the solid particles. So solid particle of a substance without forming a solution, that is very important. So without form a solution. This is the definition. Without form a solution. This is the definition for a imbibition. So absorption is what is taking place by the solid particles. Okay, solid particles. Kodai, we discuss about the stomata. We have a separate segment called the stomata. We'll discuss in detail about the stomata very shortly. Okay, so this solid particle, which is called as what the name that we give, right? The solid particle which is observing, right? This is called as imbibate. This is called as imbibate. The solid particle which is observing, that usually we call it as an imbibate. This might be a seed or it may be taken dry food. These are examples for the imbibate. Coming to the absorption of water, the water is getting absorbed by the solid particles, right? This is imbibance, and the water, right? The water is called it as an imbibate. This water which is getting absorbed is called as imbibate. Okay, this is imbibiance. Sorry guys, this is imbibance and this is Imbiate. This is imbiate. Guys, the water which is getting absorbed is called as imbiate, and the solid particles we call it as an imbibience. Okay, fine. So the absorption of water by the solid particles without form a solution is called an imbibition. The solid particle which is responsible for absorption is called as imbibance, and what is absorbed is actually we call it as ambient. Okay, co-transport and counter-transport explained. So we discuss the counter-transport that surely we go with when we're talking about the transpiration. The transpiration area, we discuss about the co-transport, the mass transport mechanism, the root pressure, everything that we'll discuss in that particular doubt. Okay, Shanmugam? Fine. Okay. So coming to the imbibition, as a result, what happened? Like the pressure. So the heat will be generated, and the pressure is also get developed. So heat is generated during the imbibition. Heat is generated. Can you someone tell me what is the name, the term that we'll give for the heat? Okay. What is the name that we give for the heat? The heat is, we call it as a heat of wetting. Heat of heat of heat of wetting, we call it as. Okay. Heat is generated because the time of imbibitions, like definitely some heat will be generated. This we call as heat of wetting. So where you could see in this case example like cellulosic cell wall. Example in case of root hairs. When root hairs absorb the water, examples. We take an example for imbibition. Root hairs that you could see, and cellulosic material like cellulosic, the cell wall and the germination of seeds. Cellulosic cell wall, cellulosic cell wall. Then you could see germination of seeds. In all these cases, these are quite a good examples that you can write for a imbibition. Here, the things and wooden doors that you could see, the rainy season, that wooden doors which is swollen and increase in size. You cannot close the door also properly during a rainy season, right? That is also a quite good example that you can make it for imbibition. I told you, like the imbibins are, you could see the seed. The seed, you can give an example like dry wood. These are the nature of like increasing in size. So you can make this also some example for imbibition. Okay. So coming to the next part, water plant relation. Coming to the plant water relation. This is the next part. This is what I studied in the textbook itself. Plant water relations. Plant water relations, we call it as. Okay. So opening and closing of stomata, all these things, we have theories. We have three Marley theory. We discuss about three important theories when we talk about this, that we'll discuss when we're talking about the stomata. Okay, fine. Plant water relation. This plant water relation, usually the interaction of what? Interaction of the plant roots. Plant roots you take in the soil and the soil water. Through how it takes place? Through the root hair. It occurs by means of through root hair. This plant water reaction, especially by means of the root hair. Where the root hair is present, this root hair is present at the root apex. This is present at the root apex, where you could see where this relations takes place, right? Where what happens, the root hairs, we know the root hairs are permeable, and also permeable to what? Permeable to water, and also it consists of pectin and cellulose. This consists of root hair, which consists of mainly the pectin and cellulosic material. It consists mainly of cellulosic material, and also this is permeable to water, so that it can absorb water from the soil. Fine. So here you could see the osmotic pressure is high or low. In this, the soil, where you could see here, the osmotic pressure is high or low. Definitely, the osmotic pressure, definitely it is more in this place, where the root hair, the osmotic pressure is very high. When coming to the pathways, you could see two type of pathway. One is the apoplastic pathway, other one is the symplastic pathway. When talking about plant water relations, we have apoplastic pathway and symplastic pathway. From this area, you can get a question. So what is the apoplastic pathway? Apoplastic pathway, where the water is getting absorbed from the root hairs. It's going to what? It's reaching what? The destination is xylem. But how it's moving? What makes the difference between the apoplast and the symplast? Where in apoplast, where it absorbed by the root hairs, is going to the xylem, but it is going through the cell wall. That is very important. Apoplast is going through what? Going through the cell wall without crossing any of the membrane. Without crossing any of the membrane, no crossing takes place. That is very important in this case, without crossing any other membrane. So it moves through the cell wall. But Symplast is not like that. Symplast, usually what happens in case of symplast, it's actually through the plasma membrane. Symplast, which usually occurs through the plasma membrane. But what happens one cell to the other by crossing the membrane? This, once it reaches the plasma membrane, there is no limitations for this one. This definitely will cross the membrane. As you see in this case, like from epidermis, right, it is going to the cortex. These are like simplest I'm talking about. So coming to the epidermis, it's going crossing the membrane, it's crossing the membrane, it's crossing the membrane, it's going to the cortex, and then it's going to the endodermis, and then it's going to the pericycle. But in this not the case for the apoplast, where it is going coming to the cell wall, it's coming this way, it's not crossing the membrane, it's coming this way, and somewhere it has to be stopped, right? Here we have something called as Casparian strip. So where the Casparian strip is present, this Casparian strip is present in the endodermal region. This based on the, this is actually basically it's what is a radial cell wall which is present in what? Present in the endodermis. And this has got some hydrophobic compound. Can someone tell me what is the hydrophobic? Hydrophobic compound which present in this Casparian stick. So this present exactly in this endodermit, and this will not allow this particular molecules to move. This will stop there. Then it should be like bifurcated, and it has to get inside the endodermis. So there is a particular compound which present hydrophobic substance which present in the Casparian strip, and this breaks the apoplast pathway. This hydrophobic substance which present in the Casparian strip, which breaks apoplast pathway in endodermis. So the compound is suberin. This we call it as suberin. Suberin is the hydrophobic substance which present in the Casparian. So that is the reason, like most of the molecules that will take, which takes the root of what? Which takes the root of symplast, not with the apoplast. Because anyhow, the Casparian strip which is going to block at some point of time, then it should be we're going to break the apoplast pathway, and this should get involved get inside in the endodermal region. So that is a reason why most of the time where the pathways which seems to be common is symplast, not with the thing. Okay, fine. So epidermis, cortex, then it's going to the endodermis. There's no such kind of interruptions in case of symplast, but in case of apoplast, it's going to the cell wall, since it's not crossing any of the membrane. But when it reaches the Casparian strip in the endodermis, WO means without crossing the membrane. So without crossing the membrane. So this makes the difference. But again, the symplast is again, there are two type of symplast, non-vacuolated and vacuolated. This can be also classified into vacuolated and non-vacuolated. Can you guys tell me what is the difference between the vacuolated and non-vacuolated? But as vacuolated is concerned, this occurs where water which will cross the tonoplast. What is tonoplast, right? This cross the tonoplast. This is vacuolated pathway. But in this case, a non-vacuolated, this will not cross the placenta. I mean the tonoplast. There is no crossing of tonoplast. What is tonoplast? Tonoplast is the outermost layer of the vacuoles that you could see. If it is a vacuolated symplast, it definitely crossed the the layer of the vacuoles, but a non-vacuolated, it will not cross over even the tonoplast of the vacuoles. By this, we can differentiate even the symplast into vacuolated and non-vacuolated. But questions come from this part, either from apoplast or symplast, or the questions which come from the Casparian strip. Okay, it should come from the Casparian strip. Okay, fine. So we're done with this part. This is about the plant-water relation. Very good. See, it's a integral membrane of the protein. As integral membrane of the protein, suberin, very good. See in the endodermal region. Okay. You have the transport of two molecules across the membrane in the same direction. So this again the pathway, antiport like symport that you did discuss, right? It's based on the movement of the molecules, right? But in case like symport, here the molecules which moves directions like two different directions. But here the main difference when we talk about symplast is it has the ability to cross the membrane. But this is not the case in case of apoplast. No wonder if it is transferring in opposite directions also, it takes place. But one thing is you should understand in symplast where the crossing of membrane takes place. But in other case the other places where there won't be crossing of a transport. So Keith, you asked like what is called as co-transport. So is it related, this mechanism related, that talking about for two molecules which is moving together. Okay, two molecules which is moving in the same direction. Can we take this as a co-transport where you just go through, where it's through both the molecules which moving in the same direction? Can we name this particular mechanism as the co-transport of the same molecules which is moving in the same direction? Can we name this as a co-transport? Is it ideal name can be give for this phenomenon where two molecules which is opening in the same direction? Fine, guys. So we're done with the plant water reactions. Next, we move on to the next part, that is transpiration. So we almost come to the next part of this chapter. This is a slide which shows, I forgot to show this one, the passive transport and the active transport. This occurs by means of diffusion, and this by means of facilitated diffusion. I told you like channel proteins and the carrier proteins that I told you, the porins and aquaporins that we discussed in this part. Then active transport, definitely we require ATP, and we have GLUT, that is glucose blood, we call it as no blood. Glucose transporters which required in case of active transport. Fine. Okay. Task I was answered for 7. Fine, guys. So we're done with hypotonic and hypertonic. So next we move on to transpiration. Coming to the transpiration, here we go. So we'll start with the transpiration now, the stomata. Fine. So transpiration is concerned. Okay. So transmembrane route is also the route that you can take place. Actually, it's not compulsorily like from the cortex like endodermis. So even the molecules which takes the route through this transmembrane route, this is also possible. Can you give an example where the molecules which move by means of the transmembrane route? Can you give one example where it takes the route that is transmembrane route? It's not the compulsory like all the molecules should go with the transmembrane route. There are few exceptions we could see. Yes, Bhaskar, you asked this transmembrane route. There is few cases where this can take even the transmembrane route. Guys, can you give me one example where the molecules even take like the transmembrane route? It's a genuine question. Fine. Coming to the transmembrane. Okay, loss of excess of water. Okay, loss of excess of water, that we call it as like transpiration. It means the loss of water in the form of vapor. Loss of water which occurs in the form of vapor. We use the term called as transpiration, and this form from any aerial portions of living, any aerial portions we talk it on living plants. But transpiration is actually where it's reduced. When it is reduced, when the water level in soil, if it is less, the water level, for example, you take in case of desert plants where the water level is quite less in the soil, that times you cannot expect that time you cannot expect where transpiration would be more, because the water soil the level is very less, where the transpiration level also which goes down. But tonoplast, and because tonoplast which is not occurring, tonoplast, there are very few molecules which can cross the tonoplast. Okay, fine. So water level in the soil, if it is reduced, water level in soil, if it is reduced, then it leads to the reduction in the transpiration also. Leads to the reduction in transpiration. So it depends upon the water level in the soil. Fine. So there is a reason why, because by means of transpiration only, what happens, all the necessary, the water, the minerals, everything is going to get transported. So without which, like, of course, it's a necessary evil, but it is necessary. If this process is not happening, then how we can expect the cells can survive, the plants, right? So that's the reason we call it as like, of course, there's a loss of water, but this evil is necessary. So that's the reason we call it as transpiration is a necessary evil. So Bharati said water and solutes are used to combine apoplastic and symplast of the transportin. Yes, this is the place where the transportin you could see where apoplastic and symplast which can be merged actually. Yeah, you're correct. So it can be merged in those cases like the transmembrane exchanges can take place. Yes, you're correct. Yeah. Okay, you're correct. Okay, so Kodai, please tell me like the book that you're referring, because just go through the book, because we go to the NCERT books like based on that the content which is given we are explaining. Okay. So the water level increases in soil where the transpiration also get reduced. So what are the three types of, so this transpiration, so the reason we call it as a necessary evil, it's called as necessary evil. So how we can classify the transpiration? There are three types of transpiration generally we make it as. What are three types of transpiration? Where cuticular transpiration, lenticular transpiration, cuticular, lenticular, then we have stomatal transpiration. So as of now, we usually think only the transpiration takes place by means of stomata, right? But there are other types of transpiration, but majority of the transpiration which takes by means of stomata, that is like 85 to 90 percentage, 85 to 90 percentage of the transpiration, which usually takes place by means of stomata. The remaining only like by means of the cuticle and the lenticular transpiration takes place. Cuticular transpiration is very less, very good, see like 3 to 10 percentage, 3 to 10 percentage of the cases only the cuticular transpiration, because cuticular cells are nothing but it's a epidermal cells. It's epidermal cells that usually you can see on the leaves. The epidermal cells which present on the leaves, on leaves actually. These leaves are responsible for cuticular transpiration. Okay, and this is also called as peristomatal transpiration. The cuticular also called as peristomatal transpiration. And coming to lenticular transpiration, so lenticular transpiration usually like where you could see the lenticles. Can someone tell me which part of the plant where you could see the lenticles, the trees that you could see, the grown trees, where you could, which part of the trees where you could see the lenticles? Even the lenticles are also responsible for performing transpiration, but it's very comparatively, it's very less. You take 0.1 percentage, somehow only you can take this type of transpiration. Usually it occurs in the woody trees, old trees where you could see woody plants, trees you could see lenticular. Very less percentage of transpiration takes place, but stomatal, but majority of the transpiration takes place in this criteria where you could see the stomatal transpiration. Coming to the stomata, structure of the stomata, the stomata where it's open and it is closed, this mechanism you would have studied, very basic concept you're studying right from the lower classes, fine. But when you see the stomatal structure, there is a difference that you could see with the monocot as well as in the dicots. But you see the general structure of the stomata, where you could see this type of cells accompanying the stomata. One is your God cells, and the God cells you could see, and you could see the subsidiary cells. This part is called as subsidiary cells, and this area, this one is called as a God cells. And the pore, this pore which tells you whether the stomata is open or closed. This is a stomatal pore, and the few cells like that it surrounding the stomata. One is a guard cell, and the other one is the subsidiary cells. Also, you could see the presence of the vacuole, the sap vacuole. We call it as sap vacuole, and there's a presence of chloroplast that you could see here. And we have the nucleus, and this cell, outermost, this cell is called as epidermal cell. This is how the structure of stomata look like. 90 to 95 percentage. I'm not sure, Jennifer, but as per the NCRT prescriptions are given is actually 85 to 90, because if we give 85 to 90 only, we can give the remaining the 10 percentage for the cuticular transpiration. It is confirmed the cuticular transpiration is 3 to 10 percentage. We go with this itself. The stomatal is 85 to 90 percentage, and cuticular is 3 to, because I'm sure about this. Cuticular it takes 3 to 10 percentage, and stomata is 85 to 90 percentage. Okay. And sir, oxygen carbon dioxide pass through the transmembrane route. Yes, of course, that's the passage of this gaseous substance which takes place. Yes, through the transmembrane route. Okay, and there is a pathway. There's a pathway where in the transmembrane route, this exchange of gaseous substance takes place. Can you tell me, Abhijit, what is the name of the pathway where oxygen and carbon dioxide get exchanged in the transmembrane? There is a specific pathway that we term it as. Fine. Okay, fine, guys. Okay, we done with this part. You could see in case of the God cells, and you could see what is it called? The subsidiary cells. Fine. Okay, we move on to the next type, that is the structure of stomata. When you talk about the structure of the stomata, the two types where you could see the monocots as well as in the dicots. Here you could see stomata, the bean shape, the God cells that you could see, and stomata with the dumbbell shape. In the monocots you could see the dumbbell shape, whereas in this part you could see in dicots where you could see the stomata with the bean shape. But just because here it looked like the dumbbell shape like structure that you could see in case of the monocots, but dicots you could see in the bean shape structure by looking at the surface. But now we're going to discuss about the structure of stomata. So how the structure of stomata, there are three categories by which the stomata can be classified. First is based on the position. First, based on the position. Number one is based on position. How the stomata can be classified. Coming to the position, and based on position of what? Based on position and the presence of accessory cells. I told you the subsidiary cells, the God cells which present, right? Based on their position and by means of the accessory cells, how this can be classified. So these can be classified into four types based on the position. What are the four types of the stomata? The first one is anemotylic, diacytic, parasitic, and isocytic. These are the four types that you could see. I'll give an example for this one. What is anemocytic? Anemocytic, you could see where there's no presence, there's complete absence of this accessory cells. Complete absence of the accessories, no accessory cells are present in this case. Example you take in case of Malvaceae family, where you could see this type of stomata. You could see in case of Malvaceae. Diacytic, where you could see two accessory cells which are present. Here, AC means accessory cells. These two accessory cells which are present, perpendicular to each other. This present, perpendicular to each other. Guys, please don't share unnecessary things because the live session is going on. The live session is going on. Please don't have unnecessary chats here. Okay. Okay. It's not going to bother anyways. Please listen to the class. Okay. Fine, guys. Please listen here. So diacytic, two accessory cells where you could see it's like perpendicular to each other. Okay, perpendicular to each other. Then you come to the parasitic. Parasitic where two accessory cells are parallel to each other. Here also you could see two accessory cells are parallel to each other. This is parasitic. CYTIC. Okay, it's not SITIC, it's a parasitic. And isocytic. Here you could see three accessory cells are present. This is an anisocytic. You could see in this case in a solanum. Example, you could see solanum species. Solanum. Coming to parasitic. Parasitic examples that you can give us a rubiaceae. Rubiaceae for this one. Example, rubiaceae. From this area you can get a question. They give us actually option. They ask you to identify what type of the stomata. Then diacytic, you could see Acanthaceae. Example, the family Acanthaceae. Okay, so based on the position of the accessory cells, you could see anemocytic, diacytic, parasitic, and anisocytic. Okay, this is all based on the presence of the accessory cells. This is the first type of classification. Second type of classification is distribution and the position of the stomata. Coming to distribution and position of the stomata, how it can be classified. Distribution and position. And the second one is distribution and position of stomata. Distribution and position of a stomata. Okay. Coming to this distribution, we have hypostomatic, anisostomatic, isostomatic, epistomatic, and we have astomatic. So this is the second type of classification. This distribution based on distribution of position of the stomata. Hypostomatic, where you could see the stomata which present on the lower side. So this is stomata which present on the lower side of the leaf. This we call it as hypostomatic. Then what is anisostomatic? This we call it as potato type of stomata. Mostly you could see in a potato, right? You could see this type of, and this you could see in the lower and upper surface. The stomata present in lower and both the sides you could see. Upper surface. Coming to isostomatic. Isostomatic, this is the oat type. You could see mostly in the oat, wheat, oat, wheat. These things I could see. God cells and the subsidiary cells of these are the accessory cells. The God cells and yeah, that's we talking about accessory cells. So the oats and the wheat, where you could see this type of thing, where equal on both the sides. So stomata, yes, stomata is equal. Stomata is equal on both the sides, both the upper and the lower side. Both the sides where you could see equally it is present. This type is called as isostomatic. Epistomatic we could see only in the upper surface. You could see in case of lily plants, example, stomata you could see only in the upper surface. Only in the upper surface you could see. Upper surface you could see. Astomatic where you could see the stomata is absent. Here the stomata is absent. So based on the distribution and the position of the stomata, this classification: hypostomatic, anisostomatic, isostomatic, epistomatic, and estostomatic. So what is meant by so which part you're asking, like 10 to 40 mu? Which you're talking about, any wavelength? In what aspect you're asking? Can you please tell me? Fine. So fine, this data. And third classification is based on the opening and closing of the stomata. Based on opening. Third classification is opening and closing of, based on opening and closing of the stomata. Okay. Okay. So guys, this we have the last 5 minutes and I'm going to close it. Hypostomata, stomata which is present in the lower side. Anisostomatic, potato type, where the stomata present in the lower upper surface. Isostomatic is oat or wheat, where you could see this type of stomata, where stomata is equal on both the upper and lower sides. Epistomatic, present lily, where stomata we could see only on the upper surface. Astomatic, where the stomata is absent. Fine. So last part is the opening and the closure of the stomata. We have the lucrine, potato, and barley type. So we have three types: lucrine type, potato type, POT potato, and the last one we have a barley type. Okay. Coming to the lucrine type, we could see in the alfalfa open all the day. The pea plants that you could see, the stomata is open all the day. Alfalfa, you could see, all the day the lucrine type of stomata which is open. Okay, open all the day. Only night time it closed. You could see the peas and beans. Then you could see the potato type open in the day and night. So it is open both day and night. This type is open day and night, but it closes only few hours in the evening. Just only few hours in the evening. Okay, it closes actually. This type of stomata only in the evening. Okay. Then comes to barley type. It only just open only few hours in the day. Few hours it opens in the day, and all the remaining type, it closes. Example for barley type, you could see wheat, the barley. In all the cases, just only few hours it opens in the daytime, and remaining times it will be closed. So guys, we almost come to the end part of this session, guys. From this area, the transportation plans experience, transpiration area, you get a questions. On the whole, you get 3 to 4 questions. Maximum 4 questions can be asked. It's almost be done. And we have very few theories. We have 3 theories that we're going to talk about: the transpiration pull, and ascent of sap. And this, the lagging areas of this particular chapter that I combine together. We combine everything. Fine. And one particular session I try to complete, because we cannot complete this plant physiology tomorrow, because we start with a new unit. That because you asked for the taxonomy of the plants, we'll start with a new unit. The pending areas of this particular chapter, like transpiration pool, the theories of transpiration, and the ascent of sap, and water potential, how to calculate the deficit, all these things that we'll discuss in a separate part, guys. So the separate session that we'll discuss under the same module. So that will do it. So I think we're done with the session, because we have only 2 minutes to wind up the session. So almost we're done this unit. So this particular unit is concerned the plant physiology. Please give more importance, guys, to photosynthesis, respiration, mineral nutrition. And from this chapter, you give importance for stomata and the transpiration. These are the common areas that you can get a question. Plant growth and development, you get questions from the hormones especially. So give importance for the hormones. So I hope you listen to this class, and so what are the test papers that you're receiving right now, just please follow those questions and keep practicing instead of sitting and listening to the class. And one more thing, guys, please go through the textbook before itself, so that whatever the information that we are providing, it will be like consolidated and you can easily understand. So thank you so much, and really appreciate your time and patience. So the timings for the class will be updated to your groups. Thank you so much.